Good evening, everyone. I think we're going to start. We gave 10 more minutes for people to enjoy a little bit of the food that's outside. It's a pleasure and an honor for me to be a moderator and a speaker to this evening's session. Michel Tremblay is very apologetic. He had actually to go to a board meeting where he's representing the Goodman Cancer Center. This is very important for donation. Il est désolé, il est réellement désolé de ne pas être avec vous et en fait ça me donne le double plaisir d'être modératrice de cette soirée ainsi que speaker in this evening. As you see, this is potentially a bilingual moderator session. So whoever, can I start by asking, are there people who speak only French here? And if that's the case, could you raise your hand? Oui, mais c'est aussi un moyen. Je vais poser la question des deux côtés. Est-ce qu'il y a quelqu'un, est-ce que dans l'audience, il y a des gens qui ne parlent que le français? Très bien. Mais de toute manière, sachez que vous pouvez poser les questions dans les deux langues, en français ou en anglais, et ça me fera un plaisir au moment des questions de traduire vos questions pour les intervenants ici. What I was suggesting is at the question time, if there were questions to be asked in French, it would be my pleasure to translate them. So I have the honor of introducing myself and the two speakers. I am Dr. Nada Jabado. I'm a pediatric hematologist-oncologist, and I work at the Children's Hospital, where I see and treat patients with cancer, but I also have an active research lab on brain tumor in children. It's an honor for me to introduce Janusz Rak, who works also at who is a Jackal chair and works with me at the Children's Hospital on angiogenesis, sur l'angiogenèse et les effets de l'âge sur la circulation sanguine in patients. And Dr. Peter Siegel, who is located at the Bellini and is a member of the Goodman Cancer Center, like both of us are, and works on metastasis and more precisely on bone metastasis due to breast cancer. All three of us tonight will try and tell you why metastasis and angiogenesis, two fundamental processes, are important for our understanding and to better treat cancer. So, we started those sessions that are very, very important by understanding. First, we start by learning the alphabet. What are the things that can promote cancer? This was actually the purpose of the first session, for you to get a little bit of the alphabet that we use. The notion of a gene, of the epigenome, how a cell is structured, and how actually those structures can can cause cancer. So this was the purpose of the first session, where you learned how a cancer could come from a normal gene that would become an oncogene, meaning a gene that can promote cancer, or from the loss of genes that are here as gatekeeper or carekeepers for our cell, and we lose them when we start developing a tumor or a cancer. The second session was here to help you understand why and how cancer is fed, actually. Why sugar could potentially be very important for cancer growth. How a cell needs to sustain itself and grow, and how cancer can actually disrupt a lot of those things that we normally use to help its growth and to grow even more. What is the purpose of tonight's session? Tonight's session is actually, for me, one of the most interesting. Everything is interesting and everything is important in cancer care. You cannot take care of one part and forget the other one. It's a continuum. And tonight's session is part of this continuum. What we're going to try and help you understand with us is how cancer cells are fed and how this feeding actually is essential for their growth and will promote actually their spread to other parts of the body. So I think an image sometimes can convey a lot of words. This is represented, it's a drawing that represents a cancer. This is a cancer, a tumor that's growing. What you immediately see is that you have a lot of things that are here colored in purple. Those are blood vessels that are essential actually to bring all of those nutrients you heard about in the second session to feed this And actually, this feeding is accompanied by cells that start to migrate, to go within, and this is also has the potential to have the tumor spread. And Dr. Janusz Rak is going to tell you a lot about the angiogenesis, and I'll go about this term later on. And Dr. Siegel will speak to you about metastasis. And my role would be to tell you how those processes could be targeted today and how they differ in children. So let's start by defining what the term angiogenesis means. We, in science or in medicine, we use a lot of Greek. So angio is for vessel, genesis is for the creation. So it's the creation of new blood vessels. Why are they important? This is a very, very, very important physiological process, meaning that you need it. You need it to become what you are. For an embryo to develop, this is a major process. And you need it actually when you heal. Whenever we have, for example, a scratch, a wound, this is a process that's mandated for the healing and a healing process to occur. So this is a process that is absolutely essential for you. All of those nutrients that you take, with the things that we eat, if we want them to go everywhere in our body, we need vessels that are like circulation to bring all of those nutrients. And we also need them to help us repair what we damage during our daily life. So why is angiogenesis so important to cancer? Angiogenesis actually is one of the major processes that cancer needs in order first to establish. You cannot establish if you don't have blood supply, and for it to grow. And those are the two things that Dr. Rak will hopefully convince you are very important to study and to try and stop. And the metastasis. Metastasis is also another Greek name. Meta is the next or distant. Stasis, it's to stop. So it's a cell that goes elsewhere, stops, established elsewhere. And this is actually one of the major problems in cancer today. Why is that? I like to think that medicine and even science go in leaps. It could be quantum leap. It could be a small little step. Every step is a gain step in understanding and curing cancer. So metastasis is not something new. We knew about it actually in the late 90s when a doctor, a pathologist by the name of Paget, described on autopsies done on women and other people with cancer that actually the tumor was not only located within the primary site, but also went elsewhere in other organs. The other thing that he mentioned in this finding, this seminal finding that dates actually in 1889, was that potentially there was the influence, this was not a random process. After that, we had to wait for a long time because a lot of things came in between before new ideas and new concepts emerged. And that was through Judah Folkman, who actually was the first person to presume that vascularization and angiogenesis were important in cancer, and Dr. Rak will tell you about that. And Isaiah Fiedler, another major scientist, who took again the seed and soil and then described the metastatic cell as a decathlon athlete, and you'll understand through Dr. Siegel's presentation why is that. So why is metastatic disease such a challenge? Well, we know how to cure cancer. We're getting how to cure a lot of cancer. We're not there to cure 100%, but we're getting there slowly but surely. And I hope that tonight's message will help you understand that we are truly on the right track and we're close. But still, 90% of patients that will die of cancer will not die of the primary tumor, but will die because it has spread. And once the spread has occurred, it's very hard to treat. So this is why the onus today in clinical medicine, but also in science, is to understand those processes that lead to the cancer spreading. And once it has spread, how better to target it. So when I say it's often intractable, unfortunately, this is the case. It's changing. If we look 20 years ago, if you had a breast cancer that had spread to the bone, your chances of surviving 5 years after your diagnosis were close to 0. Today, even though the numbers are not very high, 30 to 40 and soon 50 and hopefully 100% of those women that have metastasis, meaning the spread of this cancer to their bone or to the other organs, will live. And we are slowly changing cancer and the spread of cancer into more of a chronic disease. And soon we'll probably cure it. But as of today, this is a very difficult situation, and the onus is now on trying to prevent this situation from happening. So why is that important? Well, you tell me, for example, if you have a breast cancer, you have a tumor that's 2 centimeters, small tumor. This is the size where you could potentially detect it doing a mammogram. Already this tumor is too big because it has spread. Spread of cancer is not associated with size. You need to understand that with very, very, very small tumors that are not detectable today, this cancer will have spread. La taille n'a aucune importance, même pour des tumeurs infinitésimales. Le potentiel que ces cellules anormales qui étaient dans le sein ou ailleurs migrent et établissent est là. So anybody asks themselves, so why people, when you have a complete resection of the tumor, are urging the surgeon to look at lymph node and to stage? Because this is one of the ways for us to know if the cancer has spread. And actually, we're basing our treatment today through this staging. And women that cannot only receive, or any person with cancer cannot only receive a resection, they will receive what we call adjuvant treatment, because in some instances we know for a fact that even if we cannot detect them, the cancer has spread, and we need to treat the spread before it comes into a full-blown metastasis, meaning a growth elsewhere. So those are notions that are very important for you to understand, that even that we see, if we don't see them, they are there. And this is why the onus on putting adjuvant treatment very fast and trying to detect them with better means. So early detection is one of the major challenges, even with a very high resolution imaging technology, and we have them, MRI and PET scan, and I'll go over this. This is the image of what we have as state-of-the-art in cancer care. This is a PET scan. PET is positron emission tomography, and this is the PET part of it, and this is the CT, which is imaging, radiology imaging part of it, and this is the combination of both. Actually, this is a beautiful technique that gives you an image as if you're seeing the individual in itself. This is a normal PET. What you see, what the PET takes, anything that's very active, that meaning is requiring a lot of glucose, a lot of energy will light up. So of course, hopefully, our brain is very active and is always active. This is why the brain is always very black. It has a lot of activity. And because you inject a tracer, you could see it coming a little bit in the heart, because this is where you have a lot of blood circulation, and in your bladder, because this is where you're going to evacuate it. Everything else, and you have to take my word for it, is normal. But this is an individual that potentially has had a cancer that has spread. And with the imaging technology that we have today, we're able to detect metastasis, but only when it is full-blown. And this is the image of a PET scan, where you could see all of those black spots that are over here within the bones of the individual, all over the bone. And this is actually what it looks like when a cancer is full-blown and has spread. So what are we going to see tonight? And this is going to be Dr. Rak's talk today, is how at the primary site and at the metastatic site, providing new blood vessel angiogenesis is very important into helping cancer grow and helping cancer spread. And what Dr. Siegel is going to be telling us about is how the spread is done. Is it random, non-random? How does it do? Why? What does it take? And what do we know about this metastatic process? So those I won't go, and I'll leave that to them, is what a cell has to do in order to go and spread. Many steps, and can help. So some question that you could potentially ask, and I'll help you ask some if you don't want, because I have some burning question for Dr. Rak and for Dr. Siegel, is potentially how can we better detect those cells? How can we target them better? What are we doing to improve our treatment, and what is this knowledge translating into? So I'll leave the ground for Dr. Rak, and thank you for your patience.
Can you hear me? Not yet? Oh, what about now? Is it better? How about now? Is it good? Okay. Oh, thank you so very much. That was a lovely introduction. I'm not really planning on giving a talk. I just wanted to share with you a couple of comments that would expand on what Dr. Jabado so eloquently described, and maybe bring up some of the ideas that emerged in biomedical sciences around the issue of interactions between cancers and blood vessels. And I think it probably is fair to say that really any progress ultimately has its source in ideas. So what I'd like to tell you about is a little bit about blood vessels, how they function as supply lines of the tumor, escape routes of metastasis and therapeutic targets all at the same time. So I think it's probably trivial, and I don't have to really stress this too much. It's trivial to say that blood vessel system, or circulatory system as it's known sometimes, has a very unique role. So we are all composed of a variety of cells and organs and organ systems, but one organ system stands out as a particularly distinct in its role, and that would be the blood vessel system. Why is that? This is the system that, in fact, connects every cell in the body, or every part of the body and every cell in it, to the outside world. And this is through the intake of oxygen and nutrients from the outside and distribution of these respective components throughout the body. And also, the vascular system connects the cells to each other, and this is through hormones and metabolites that constantly circulate. So it's probably important to keep in mind that although there are tissues in which there's more or less or different blood vessels, every cell in the body is usually no more than 100 microns, or 1/10 of the millimeter from the nearest functional blood vessel. So every cell has that functional connection. And that's a very consequential situation, and of course important from the point of view of different functions, because different organs performing different functions in the body actually have very different vascular systems. So vascular system is not some kind of a random bundle of pipes through which blood flows freely. It's a very structured and very purposeful system. And so, for example, in the heart, we would have this kind of architecture. And in fact, what these images are, they're so-called corrosion plastic casts of blood vessels. So what one does is one takes an organ, let's say the autopsy or from experimental animals, and fills it with a plastic resin and lets it harden. So then, of course, the old soft tissue can be digested, and this will give you a rather accurate reflection of all the architecture of the vascular system in particular organs. So you can see that there are different architectures in the heart, in the kidney, in the brain, of course, in the liver, and also in the gut. There are very different patterns of blood vessels, and each of them has certain particular geometry to it. And this geometry reflects both the architecture of the organ and its function. So, for example, in the intestine or the gut, blood vessels are structured in such a way that, that they surround. The so-called intestinal villi from which the nutrients are being absorbed. And so, of course, I just wanted to say that blood vessel growth is very controlled. So in order to achieve these sorts of architectural constraints, the blood vessels have to be controlled, and their growth has to be very tightly regulated, and their integrity is also controlled from within by the so-called coagulation system or blood clotting system. So whenever blood vessels are disrupted, the clotting system of enzymes contained in the blood kicks in and plugs up the hole. And of course, we can study some of these things in mice. So, for example, one can generate a mouse that has green glowing proteins in it, or either ubiquitously or in specific organs, or including blood vessels. So one can then trace blood vessels in different organs and visualize them, whether they resemble the human counterparts or not, and what happens to them in pathology. So this is how we can do some of the research in the surrogate setting, whereby we can use experimental animals to answer some of the burning questions that may arise. And of course, the key question of this evening is, what happens with this orderly architecture? How is this architecture really related to cancer? And of course, when in a particular organ tumor occurs and, of course, metastasizes throughout the body, it is hard to imagine that this would not have a consequence for blood vessels, and it does. So, for example, this is the same organ, the mucosa of the intestine, in which tumor has developed. So, as you can see, there is really no comparison. This is a site of chaotic, crazy outgrowth with disruption of the architecture, with many vessels, many more than necessary, and in wrong places, with structures that normally don't exist in a normal organ, and with all kinds of abnormalities that make this particular system of vessels very distinct, not only architecturally. This is not just that they look different. They are different. They are very different in every possible way you can imagine. So not only they are different, but they remain, oddly enough, necessary. And why do we know that they are necessary? Because when we take a cross-section through the tumor tissue, for example, and we label blood vessels with certain dyes, we can observe that tumor cells essentially fall into two different categories: the ones that live, and those are usually surrounding the existing blood vessels, and those that die, and those are the ones that have been removed or pushed away from blood vessels. They simply cannot sustain their growth capacity at a greater distance from the blood vessel than is required for them to absorb oxygen, nutrients, and growth stimulatory factors. So blood vessels are both abnormal and very necessary for tumors, and we can, again, observe these sorts of processes by using experimental animals. For example, this is a green mouse that I showed you before, and we can see that in the tumor of that mouse there's all kinds of mouse or host-derived cells that either form blood vessels or form different structures within. So these so-called normal cells play a big part in the tumor. So tumor is not just a collection of cancer cells. Tumor is a complex structure that has its own systems of laws and regulatory processes. And this is an important statement, and it leads us to the question, what really happens here? Why are blood vessels so abnormal? What really triggers this whole chaos that I just showed you? And it turns out that tumor cells may exist in the tissue, and then, for example, this is idealized tumor cell somewhere lost in the tissue, as far away from the nearest blood vessel. And yet these tumor cells, at some point of their natural history, they could normally exist in that state, we can assume, for extended periods of time, and nothing would happen unless and until something triggers these cells to become activated. And they can be activated either through stresses, different kinds of stresses they can be subjected to, mechanical stresses, stimulation with different proteins, or intrinsic changes within tumor cells, as signified by the oncogenes Dr. Jabado mentioned. And when these stresses act upon tumor cells, they become alive. They change fundamentally their relationship, which is initially relatively indifferent to the neighboring blood vessels. And they change this relationship through a number of SOS or emergency signals they emit as a result of the stress. And one of these signals is called vascular endothelial growth factor, VEGF. This is just a protein that is designed in the body to facilitate the talk between the cells and the vessels. So this is the main message that cells can send to the vessels. But there are also other messages. There are other angiogenic, so-called angiogenic factors. There are small multi-molecular complexes from the tumor's plasma membrane called oncosomes. So there's a number of ways by which tumor cells can actually send away the signals towards the neighboring blood vessels. And when they do, things become really ugly. So initially, these dormant cells lie silent somewhere in the tissue, and when they start producing angiogenic factors, blood vessels, as good soldiers, they emit their own projections into the tumor neighborhood, vicinity, and these projections, vascular sprouts, start growing and growing, and until they cause a nuclear explosion of tumor growth, which essentially takes over, and the tumor starts becoming aggressive, and then, as Dr. Jabado has mentioned, begins to metastasize. And of course, this is a very interesting process, and of course, it will be very gratifying intellectually to study this, but this is not why we do it. Why we do it is because this process has very significant therapeutic consequences, and these consequences were the first time appreciated about 40 years ago by this unassuming gentleman by the name of Judah Folkman, who was then a not much respected pediatric surgeon in the Harvard Children's Hospital, told to stick to his surgery and not take too much interest in tumor biology, but he persisted, and he in fact laid a foundation for a completely new way of looking at cancer control or cancer treatment, and namely this was built on the premise that on the question rather, which can be phrased as follows: can cancer cell or cancer cells be controlled from the outside? So are we only left with instruments that could kill cancer cells or control their interior, or can we actually control their behavior from the outside? And one way in which he envisaged how we can control it from the outside was by the following observation. It was known that certain types of cancers may lay dormant. So there are cancer-looking cells in organs, and yet nothing much happens except for these cells being there. And this can take many years until something happens within these cells, as I mentioned earlier. The change that occurs within these cells around them, which brings about the process I described a minute ago, which Folkman called angiogenic switch. And of course, this program, as I mentioned, triggers an ingrowth of blood vessels into the tumor vicinity. The tumor begins to grow, and so on. And this is a very nasty and unpleasant and dangerous process. But as every nasty, dangerous, unpleasant thing, this also presents itself as an opportunity in terms of therapeutic intervention. Namely, one can think of at least two different ways to block this process, namely by blocking the angiogenic switch and by blocking blood vessel response to the angiogenic switch. And what you end up having is possibly, as he theorized, a return to a dormant phenotype or the dormant state of cancer. And of course, this was a theory in 1971. There was absolutely no support for this whatsoever. And, he and many others have spent many years to actually study this. Now, the key question is, was he right in saying, or are people right in saying that tumor cells may lay dormant in the tissue? Okay? Because this is the basic premise of all this theory, and of course it's been long known that certain cancers occur with a relatively low frequency. For example, thyroid cancer. What is the chance of getting a thyroid cancer? Very low. It's below 1%, perhaps even less than 0.1%. So a group of pathologists have published a paper in a rather prestigious journal in early 1990s where they took patients who died of other causes, car accidents, cardiac arrests, and so on and so forth. And they actually tested how many cancer cells, or would they be able to locate or identify cancer cells in different organs of these patients. And for thyroid cancer, for example, which is, again, a very infrequent type of cancer, they found that 100% of patients between ages of 50 and 70 had cancer-like looking cells in their thyroids, and yet they would never die of these cancers. So you can have a cancer, but you don't need to die of it. That's basically the message. And I think this is so why is it that the cancer cells are there and there's nothing, the disease doesn't emerge? So one possibility, of course, is that these cells are not capable of triggering blood vessel growth and angiogenesis. And of course, if that was the case, then we can consider the following possibility. So this is the angiogenic scheme that I showed you about. There's vascular endothelial growth factor, the angiogenic factor, which is produced by tumor cells, and blood vessels are coming to it. What I didn't tell you is that this whole process is actually influenced also from a distance. So bone marrow cells can be emitted from their sites, and they can migrate towards these sites where blood vessels are stimulated, and they can actually support and stimulate further growth of blood vessels in the tumor or around the tumor. So this is a somewhat more complicated process overall. And knowing this presents us with at least a couple of different possibilities to test the question whether we can block angiogenesis and thereby block the tumor, or is it possible, is it feasible to intervene in such a fashion? So for example, what we could do is we can block production of angiogenic factors by tumor cells, or we can block the activity of these factors. We can block VEGF, or this vascular endothelial growth factor, from acting on endothelial cells. We can also block endothelial cells from responding to this factor. And finally, we can block bone marrow cells from migrating and facilitating this process altogether. And so there's at least 4 different possibilities. And since 1998 or slightly thereafter, there's already, within the last decade only, think about this, Wright brothers have constructed a technically accurate airplane and flew it in 1903, right? And the first commercial flight has taken place in sometimes in 1950s. So it took half a century to take Wright brothers' airplane and make it into a product that was actually usable. And in only 10 years, or slightly more than 10 years, there's already several. This is just a tip of an iceberg. These are all approved, clinically used drugs that target different aspects of angiogenesis. So for example, the drug you may have heard about, Herceptin, blocks certain tumor cells from producing vascular endothelial growth factor. It does other things as well. The first ever approved anti-angiogenic drug, Avastin or bevacizumab, approved in 2004, it's an antibody against vascular endothelial growth factor. There's a number of agents, Sutent, Nexavar, Votrient, which block endothelial cells from responding to this very factor and from other factors. And finally, Avastin also acts to some degree on bone marrow cells. So these drugs, anti-angiogenic drugs, are already in existence, and hundreds of more of these drugs are being looked at and tested. Now, of course, I don't want to leave you with the impression that this is all case closed and so on. There are problems. Every solution creates problems. So some of the types of cancers, for example, and we don't know why that is, are particularly sensitive to this treatment. So for example, anti-angiogenic therapy over the last 2-3 years has fundamentally changed the way kidney cancer is being treated, especially metastatic kidney cancer. It also had an impact on liver cancer treatment. It is beginning to make a dent in brain tumor treatment, although this is still in its infancy, and has a significant role in treatment of metastatic tumors in the gut or colorectal tumors. However, there are tumors in which this treatment has absolutely had no effects whatsoever until now, and some of those include pancreatic cancer and some of the forms of skin cancer, particularly malignant melanoma. Why? We don't know. Well, we know some of the answer, but not all of them. There's still more whys or more questions that are being looked at. And for example, some of my favorite whys are, why is it that not all cancer patients respond equally? Even within the same type of tumor, there are those who would benefit more and those who would benefit less, and we need to understand why that is. Why many drugs active in mice, for example, in preclinical testing, eventually fail in the clinic. Many of them do. Why there's no approved anti-angiogenic therapy in children with cancer. This is a very painful statement for somebody like myself and Dr. Jabado, because we work in a pediatric hospital, and we would really like to to see this change. And why do children seem to respond differently than adults to these drugs? And I would like to focus for a few minutes on this last question because this is something that speaks to our current interest. So one thing that I would like to mention, which is not widely appreciated, I have to say, and you wouldn't read it in textbooks and probably not on the internet, is that vascular system actually ages with us. Okay, and as it ages, it becomes prone to its own diseases. One of these diseases is called atherosclerosis, which we all know exists, and many of us here have it. And I would say probably a great many of us would have it because after the age of 40, if this red line is to some degree representative of the progression of this vascular disease called atherosclerosis. And as you can see, the frequency is relatively high. This is the percentage in the population who have this particular manifestation of atherosclerosis, that is plaques in carotid arteries, and there's a very significant percentage of those of us who are older than 20, I admit I am, who have that particular condition. It's hard to believe, but that's true. So just think about it for a second. So if you, and this is the cancer incidence. So in a kid who is 5 years old, essentially this is a non-existent condition. So the vascular system is very different here than the vascular system of a 70-year-old gentleman who develops, let's say, prostate cancer. So we are dealing with a completely different target or different system to manage at different ages. Okay, so pediatric blood vessels are different than geriatric or middle-aged blood vessels. That's basically what I would like to mention. And in fact, is this true? I mean, this is not just a speculation or some kind of conjecture. This is our own study where we simply took a group of patients with kidney cancers who have been successfully operated and essentially cured of kidney cancer, but they would differ in terms of age. So some of them would be older than 65, some of them would be younger than 65, and the range is relatively wide, between 30 and 65 in this case, and between 65 and 85 in this case. And as you can see, older patients have different frequency or densities of blood vessels in their tumors. This is the same tumor, but there's different numbers of blood vessels per tumor depending on age. So aging is very important. I would like to see at some point a children analysis here, but the data is not available at the moment. Now, this brings us to perhaps last question I want to touch on. You know, with all this aging talk, is it a valid question? Could we ask the question, could vascular aging influence anti-angiogenic therapy? So since the target is different, blood vessels are different with age, does it matter? And I can't offer you any clinical data because I don't have any. They don't exist. And this may be something that we might be able to contribute in the future. But I can offer you some mouse medicine data, and this is what it is. So these are three groups of mice. This is a pediatric group of mice. This is a middle-aged, with middle-age crisis group of mice. And these are geriatric mice, which have atherosclerosis. And they're injected with the same tumor. So it's identical tumor from the same tube. They're being injected with the tumor, and they develop this tumor. And these bars signify how fast tumor cells grow. Okay? And as you can see, in pediatric mice, they grow pretty fast. In middle-aged, they grow not so fast, and they grow the slowest in geriatric group or old age group with atherosclerosis. Now, what happens if we superimpose on this a protocol of anti-angiogenic therapy that is different than I mentioned? There's this form of anti-angiogenic therapy that is essentially based on giving anti-cancer drugs at a particular, according to a particular regimen. So we give these drugs at very low non-toxic doses, but very, very frequently, like daily. And so what happens if we, and this works as anti-angiogenic, this blocks blood vessels, doesn't block tumor cells. So what happens when we give these mouse patients the drug according to this protocol? So that's what happens. In the young mice, in the pediatric population, this actually works pretty well. In the middle age, it works still, but not so good. And in the geriatric population, there's essentially no effect. So as you can see, this is the same tumor and the same treatment, and yet it has a very different impact depending on the state in which vascular system is at the time of treatment. So we need to understand these things. This is just an example. We need to understand what is going on to be able to use these agents and these principles more effectively. And also aging affects metastasis, as we know. So basically, I just wanted to finish up, and potentially, hopefully, provoke some attacks on me from you or questions. So what I did try to tell you is the tumor growth depends on blood vessels. Blood vessels contribute to cancer invasion in many different ways. We didn't elaborate on this, but maybe Dr. Siegel will tell you about this. Blood vessels deliver many things, such as anti-cancer drugs, bone marrow cells, immune cells. Numerous go and stop signals to the tumor. So there are highways leading to the tumor, and they deliver all this information. Metastasis is defined by blood vessels at several levels. Blood vessels are conduits of metastatic cells, as Dr. Jabado mentioned. Blood vessels provoke bone or mediate bone marrow cell involvement in metastases. They develop oxygen and nutrients to metastatic tumors as well as the primary tumor. And also there are sites of clotting. Blood clotting is a factor in metastases. Blood vessel-directed agents are active in cancer. I hope you believe me. And certainly FDA is of that opinion because some of these drugs are already approved. Circulatory system is complex, and so is its involvement in cancer. And therein lies not only problems, but also opportunities, which we are beginning to explore. And developing better anti-cancer treatments depends on our understanding of tumor blood vessels and their mysteries. And this is basically the main point I wanted to make, is that we are just cutting into it, and perhaps this is relatively early days, but already there are some promising signs, and hopefully they will continue. I would be glad to know what you think about this. Thank you so much.
Our next speaker is, or the person who's going to speak about metastasis and bone, is Dr. Siegel. So I just, while Dr. Siegel is setting up, I want to tease a question out of you, Janusz. Just, this is a foreplay before we pass. So you're telling me that potentially children might benefit more than adults from those treatments that are directed against blood vessels or new blood vessels, and that they're not getting it. This is what you're telling me.
Well, that's one possibility. I would say that they will have different responses, and therefore. So now he's asking me whether or not children would benefit more. I would say that's a possibility. There is a possibility, which I think is unused, that children may actually respond better to certain anti-angiogenic treatments, but it is even more likely that they will respond differently. And I think part of our program that Dr. Jabado and myself and others at Children's are trying to develop is to develop ways to devise drugs that would be specifically tailored to children. Because we believe that these agents that are being used in adults, they may have their role, but there may be other possibilities that should be looked at, and perhaps there could be pediatric-specific agents or pediatric-specific approaches could be used. I hope that answers your question.
Okay. Well, good evening. My name is Peter. I'm an assistant professor here in the Goodman Cancer Center, and my lab is next door in the Cancer Pavilion. And what I hope to convey to you tonight is maybe a better understanding of the metastatic process as it relates to breast cancer, which is the disease that is of particular interest to my laboratory. And I want to take you through sort of the general aspects and the process of metastasis and how we try to better understand this process in the laboratory. And to develop possibly new therapies or new methods of tackling this particular aspect of the disease. And so as Nada mentioned, metastasis really means a word that we use to describe the process of metastasis, or of cancer spread. So metastasis really is the process by which the cells within the primary tumor are spread distally and arrive at locations in the body where they can establish and grow as secondary tumors. And really the problem with metastasis is it really marks a transition from local disease, which is confined to the primary site, to a systemic disease, in which now we have secondary colonies of tumor cells growing in tissues such as the bone, the lung, the liver, or the brain. And so it marks a transition from the ability to manage the disease locally by surgical intervention or local radiation to a problem now where we have tumor cells deposited in multiple locations in the body. And why is metastasis so important? As Nada mentioned, really it's the development of this metastatic disease that really is the life-threatening transition in terms of disease progression. And what we can see is that in cancers that remain confined to the breast, the survival rate for patients for 5-year survival is very, very high. And that these carcinoma in situ, which are small benign cancers within the primary site that are fully bounded by a basement membrane, these lesions can be effectively treated by surgical removal. However, as this disease progresses and we begin to see tumor cells actually breach this basement membrane and begin to invade locally and spread to the lymph nodes, the chances of surviving the disease begin to drop. And when you get to the stage 4, where you actually have metastatic dissemination throughout the body, the percentages of patients surviving for 5 years after their original diagnosis drops to about 26%. So it really is the appearance of metastatic disease that is the life-threatening component of the disease. So how do tumor cells escape their primary location? And this has been touched on by Janusz in the first talk. And there's 2 main ways for which tumor cells can escape the primary site and spread throughout the body. And it uses 2 circulation systems within the body. The one is the lymphatic system, which is responsible for draining interstitial fluid from tissues and returning that fluid back into the circulation system. And the other is the blood circulatory system that Janusz described so well in the previous talk. And both of these systems are avenues in which the tumor cells can, once they've obtained the ability to migrate locally, can move through the tissues and intravasate into either the lymphatics or into the blood vessels. And when it takes a lymphatic route, these tumor cells can eventually make their way back into the venous circulation and be disseminated throughout the body. So these are the 2 main principal routes by which tumor cells can begin their distal dissemination. So what do we mean by the metastatic cascade? And Nada mentioned the analogy of a tumor cell being equated to a decathlete. And that is because there's so many different processes that are involved in the metastatic cascade, which are outlined here. So this first 2 steps is really the growth of the lesion in the primary site, the angiogenic switch that Janusz described, in which these tumor cells must instigate the recruitment of blood vessels to supply the growing tumor. But it also provides an avenue for these tumor cells to disseminate. And these tumor cells then must sort of intravasate or break their way into the blood vessel. And this process is dependent on host cells as well as the tumor cells. So this is sort of a message that will come back over and over again, is that we're not really just dealing with the transformed cells themselves, but how these tumor cells interact with stromal cells within the tumor microenvironment, with host cells that facilitate each one of these steps, both at the primary site and at the metastatic site. So once these tumor cells have gained access to the bloodstream, they must survive while they're in circulation, and they then arrest in a distant capillary bed. And there's a number of different hypotheses about how these tumor cells, once they stop in a distant capillary bed, how these cells will actually get out into the metastatic tissue. And this is a process which is typically called adherence, where you have to have the tumor cell interact with the endothelial cells that line the blood vessel and extravasate or break through this endothelial barrier and arrive into the parenchyma of the metastatic tissue. And then really there's a decision that occurs whether that tumor cell, once it's in the metastatic site, whether it's going to grow or whether it's going to die, or whether it's going to stay there as a dormant cell, as Janusz was describing. And it's really the ability of these tumor cells to respond to and influence their microenvironment once they're in the metastatic site, which will dictate whether they'll actually progress to a macroscopic lesion that becomes clinically detectable. So these processes have been described as both efficient or inefficient, and there's some debate as to whether these steps in the middle are really efficient steps or not. A lot of this has come from the use of animal models to assess how well tumor cells are able to accomplish each one of these different steps. But really, what's clear is that a lot of the barriers for the development of metastatic lesions in the secondary site come from the ability of tumor cells to respond to and influence the microenvironment once they've reached the metastatic site. And this is really believed to be the point in the metastatic cascade where a lot of these tumor cells that disseminate from the primary are killed off or die out through processes of apoptosis. So what are some of the factors that will dictate where a tumor will spread to? And there's two main ideas surrounding the ability of tumor cells to spread to distant locations. And the first is really the blood flow patterns in the circulatory system. And the idea is that tumor cells that originate in an organ such as the colon will have the highest frequency of metastasis to the organ to which the blood draining from this organ is the next capillary bed that these tumor cells will see. And that is definitely the case for colon cancer. So colon cancer metastases to liver is the most frequent site of metastasis of this cancer type. And that's because the blood flow draining from this organ goes to the liver first before it is redistributed throughout the body. So that, in certain cases, that holds true. But in other cases, such as breast cancer, that's not necessarily the case, in which you have the ability of tumor cells to disseminate either through the bloodstream or through the lymphatic system. It gets sent back to the heart and then pumped out to the lungs to become oxygenated. You would think that the lungs would be the highest or the site of most frequent breast cancer metastases, but that's not the case. In the case of breast cancer, the bone is actually the most prevalent site of metastases, followed by the lung, the liver, and the brain. So that suggests that really simply just blood flow patterns is not enough to explain where and why tumor cells will metastasize. And there has to be other explanations for this event. And this sort of comes back to what Nada was describing in the introductory slides, is this whole idea of the seed and soil hypothesis. And again, this is a very old idea. It was first proposed in 1889 by Dr. Stephen Paget, who came up with this idea that if you use an analogy that when a plant goes to seed, its seeds can be carried everywhere. But the ability of those seeds to live and grow is determined by the type of soil that they land in. And if there's fertile soil, then those seeds will grow. But if those seeds land on dry or arid ground, you're not going to have the growth of the plant. And this is a very nice analogy when you're describing the process of metastasis, where you really have to have this compatibility between the cancer cells that are spread throughout the body and the soil, which is really the site of metastasis in which these tumor cells find themselves. And so, as I mentioned, this is really the driving principle that sort of underlies our current thinking about why certain tumor cells metastasize to certain places. And again, in the case of breast cancer, you have bone being the most frequent site, lung, liver, and brain. And we can think of this as almost a selection process where you have a diversity of tumor cells within the primary site. And these cells will disseminate likely randomly throughout the body, and that these tumor cells will deposit in these various different organs. And once they're there, there's a certain number of things can happen to them. Either that cell is incompatible with this particular microenvironment and those cells will die out through a process called apoptosis. They may persist in the site as a single cell or very small clusters of cells that really can't be detected clinically. Or they can actually begin to grow again and form a secondary metastases. And the interesting question is, why is certain cells in the primary tumor suited to grow in the bone, while those same cells, if they landed in the brain, would fail to form progressively growing metastases? And we believe that this has a lot to do with the complementarity of these tumor cells and what they're expressing that allows them to both respond to and to cause changes in this metastatic microenvironment that would favor their outgrowth. And so you could sort of think of it in this way, in which you have two individuals. One individual finds himself in a very unique microenvironment that requires a certain set of equipment or tools that he must have to survive. And he'll do very well in this microenvironment. But if you were to take this individual with this set of equipment or tools and you were to place him in this microenvironment, that would have disastrous consequences, and vice versa. And so the same thing is believed to occur in metastases, where you have the outgrowth of cells in the lung, and they've been selected for their ability to grow there by virtue of the genes that they're expressing and the proteins that they're producing. And if you were to take these cells and you were to place them in the microenvironment of the bone, they would fail to thrive. And this has been borne out in using animal models to look at this particular process, using cells that have been isolated to be very aggressive to the lungs, and these cells really can't metastasize very well to the bone. So how do we go about learning what this toolbox or this set of equipment that tumor cells can use to be metastatic to a particular organ or tissue? And one of the ways that we can do that in the laboratory is to employ a mouse model in which we begin with breast cancer cells that have a very poorly metastatic phenotype. And we can inject these cells into the animal and allow them to spread to the bone. And by flushing these cells out of the bone and back into culture, we can repeat this process over and over again with the hopes of actually selecting more highly aggressive variants that can metastasize to the bone. And then what we can do is we can ask, just in a global fashion, what are the genes or the gene products that are present in those cells that are metastatic versus those that are poorly metastatic. And in this way, can we find some knowledge or gain some information about the equipment that these cells have that allow them to form bone metastases and these cells lacked. And this is a process that can be repeated over and over again using selection to the liver, to the lung, to the brain. And what this has shown is that when you do that, even when you start with the same starting population of tumor cells, you begin to pull out different subpopulations with very much different gene expression profiles, whether they come from the bone, from the brain, from the liver, or from the lung. And this is indicating that these tumor cells have gone through a selective pressure. They've been attuned to that metastatic microenvironment, and they survive there by virtue of the genes and gene products that they're able to express. So this is sort of taking a very tumor-intrinsic view of the metastatic process. And it's really describing a set of genes or gene products that are important for the tumor cell to express themselves. Another way that we can approach this is actually going to clinical material that we obtain from breast cancer patients. And this is sort of asking, can we understand what these tumor cells are now expressing once they find themselves in this new metastatic microenvironment? And the way that we do that is through a procedure called laser capture microdissection. And this is really a technique that allows us to go in and to take a piece of tissue that came from an invasive ductal carcinoma of the breast, and we can go in and actually selectively capture the individual tumor cells out of this tissue. And we can take these epithelial tumor cells that we've isolated, we can extract RNA from them, and we can look at what genes are being expressed and what gene products are being produced. And we can do the same thing if we look at patients, breast cancer patients with known bone metastases, using biopsies that have been taken and where we have a little fragment of bone with a tumor within the bone marrow. And we can actually go in again and individually isolate these metastatic tumor cells and ask what's being expressed both in the primary and what's being expressed in the bone metastatic site, and try to understand, based on what these tumor cells are expressing, what it required in order to form that lesion in the bone. And through these sort of combinations of approaches, the field has been able to learn quite a lot about how tumor cells interact with the bone microenvironment. And through this knowledge, it really has generated potential targets for intervention. And so what's really important is that these tumor cells are expressing a number of different genes or tools, if you will, that allow them to interact with the cells within the bone microenvironment. And really, it's this interplay between the tumor cells and what they're producing that allows them to influence host cells within the bone to actually destroy bone and to release proteins from the bone that then feed back on the tumor. So not only is the tumor producing factors that allow it to modulate its microenvironment, but it's also expressing proteins that allow it to respond to growth factors or cues that are coming from the microenvironment. And so in this way, these tumor cells really are having to interact proactively with these different cell types within the metastatic setting in order to establish a secondary lesion. But what's important is that by understanding how these interactions occur, we can come up with targeted therapies that are hopefully going to interrupt this process and diminish the ability of these tumor cells to establish a lesion in the bone. And one good example that recently has been taken to phase 1 and 2 trials by Amgen is an antibody that recognizes this protein called rank ligand. And this is a protein that's very important for osteoclast cells to resorb bone and to release these factors that cause these tumor cells to grow. So it's a good example of actually a therapy that's not targeting the tumor cells directly, but you're targeting this protein that's very important for a host cell type to function. And by blocking that, you sort of deprive these tumor cells of necessary growth factors that allow them to proliferate and to grow into a macroscopic lesion. So I think really the take-home message is by understanding the factors that allow these tumor cells to communicate with the cells within the metastatic microenvironment, we open up the opportunities to try to interrupt this communication and to stop these tumor cells from growing. So what I've tried to sort of cover briefly is the idea that metastatic cancer is a phase of disease that has spread from the primary tumor, and it's now become a systemic disease. That this process of metastasis is an inefficient process, and it's a non-random process, and that the ability of tumor cells to metastasize to distinct organs and tissues can be characteristic of different cancer types. The ability of these tumor cells to grow in the metastatic site really relies on a compatibility between the tumor and the microenvironment. And that, coming back to this idea that you're really not looking at the tumor cell in isolation, but you're really looking at the ability of these tumor cells to influence and change the behavior of stromal or normal cell types, and then be able to respond to those changes that occur as well. And that hopefully by identifying the genes that are being expressed by cancer cells that can metastasize to these different organs and tissues, we can uncover Achilles' heel that might be exploitable for a therapeutic intervention. Thank you.
How are you doing? Are you still with us? How are you faring up to now? Are you still up for one more? Okay, good. So I'll start actually by asking you a question. This is my way. Usually when I start speaking and it's pediatric cancer, people go. It's as if pediatric is something that interests little persons. I promise you, I will speak pediatric, but I will also try, je vous donnerai aussi plein de choses sur les adultes. So don't go away. We still have some very interesting thing to share with you. And so the question I had for you, how many of you know what is a clinical trial? Okay. How many of you know what are the different phases? What is a phase 1, a phase 2, a phase 3, and a phase 4? Okay, so I need to explain. So a clinical trial is, you saw this beautiful work done by Peter and by Janusz and by other researchers. The way it's done, you identify something, a target. You design against it a drug. This is what pharmaceutical companies do all the time. What you want to know is this drug efficient. Before you start using human beings as cobaye, hamsters, or rat, whatever, you use it in cell lines, meaning some cells that were taken from a cancer, from a patient, and where you could potentially see if you can stop the growth, or you can stop the vessels from coming, or you could actually modulate the behavior of this cell. If it is nice, you take it to the next step, which is our friend the mice. So mouse models. So we have means, and you're going to see through my talk, and you've seen already through Peter and Janusz, the mouse is actually a very powerful tool because you could induce tumors in there and then test whatever treatment you have available to see if this tumor is going to decrease in its growth, because you need to justify that it might work for us human. And you need to justify that you didn't kill the mice, or you didn't kill the rat or the hamster with this treatment, and you didn't give a lot of side effects. If all of that is fine, you go to the human, and you start with what we call clinical trials. What is a phase 1? Phase 1 is actually you take a drug. It's a new drug. You test what is, you're not looking at efficiency. You're not at all looking at efficiency. You're just looking if you could give it. What is the maximum dose you could give it, and is it safe to give it? So you're not even looking at the result for treating a tumor. You're just looking at if you could give it and safely give it, and what we human could tolerate as a maximum dose. You go to phase 2. Phase 2 trials are the same drug with a maximum tolerated dose. This is what we call in our jargon MTD. And here we're looking at efficiency. So we take patients that actually, unfortunately, are in a desperate situation because they have failed all of the available treatment out there. And we give them this drug with the hope of seeing an effect. If we see an effect, we take it to the next step, which is what we call a phase 3, which is actually comparing to the standard of care of treatment that's available out there, and seeing if it is providing an effect on a bigger scale of patients that actually are could receive the standard part of the treatment. And most times for phase 3 trials, we do it with a randomization. What is a randomization? It's like you flip of a coin, where you randomly assign either the experimental arm, where we're seeing if this drug is efficient, or you're just given the standard of treatment. And why is that? And that's important, is we don't know if this drug that seems very promising in mice will actually prove to be efficient in human. And we need to do this type of trials before we could say safely that it might give us an advantage. All of those steps are very time-consuming, very emotion-consuming. You wouldn't imagine I have to present them to families every day. And when patients fail treatment that we have to give them, I have to go and dig if there are any drugs that looks promising that are being tested, just to try. But this is how we move science, how we actually breach the bench to the bedside. And the phase 4 is your Tylenol drug that's available on the market that you could use however you wanted without any restriction. So those are what we call the clinical trial. And when Peter mentioned a phase 1 and a phase 2, sometimes they are joined. That means that we don't know if this drug is efficient. It is very promising in cell lines and in mice, in animal models, but we don't know if it will do anything in humans. But still, this is the very first step for us in providing a cure tomorrow for patients that need it. So, cancer in children. Are children very small adults? I'll try and convince you that we actually are a continuum, that we have a kind of a common core, but we have also our differences. And how can I convince you of that? Some pies. This is a pie that was taken from the European group. Europe and North America are very similar, actually. And what are the cancer at all age in females above the age of 30? As you see, a lot of breast cancer, colorectal cancer, and other cancer. If you go to males, they don't have breast cancer. They have lung cancer. Smoking is one of the major producers of lung cancer, and it affects actually equally women and men, but men smoke more than women. And the colorectal cancer and all of the other cancer. Children. Well, children, you could divide them even more. There is actually an orphan population that are the adolescent young adult, meaning patients that are between the age of 15, around 15, till the age of 25, 30, where actually the most common cause is a lymphoma, which is a cancer of the lymph nodes, a little bit of leukemia, brain, and sarcomas, which are cancers of the soft tissue and the bone. So you see, it's a completely different distribution. And this distribution is going to be even more different if you go to children between the age of 0 to 15, where you will see that there are two major tumors, which are leukemias and brain tumors. So just by saying that, you see that cancers are different. We barely ever see breast cancer in pediatrics. We barely ever see a cancer of the skin, the melanoma. We see a lot of leukemias. We see a lot of brain tumors. And even in those brain tumors, they are different from the adults, and I'm going to tell you that. So what have we done? Leukemia, we're getting to treat. We're getting better at treating it. I'm not saying it's a done deal. I'm saying that in pediatrics, one of our strengths, because the numbers are small, is that we've regrouped. We are part of different consortia. If a child is seen at the Montreal Children's Hospital, most probably the same treatment will be delivered here at Harvard, at Sloan Kettering, in the US, in Europe, the same drugs, almost the same protocols. And it's by regrouping our expertise, regrouping the tumor samples, that we were actually able to advance a little bit our treatment. Those are the cure rate, the mortality per, Those are the cure rate, mortality per million due to leukemia in the 50s. This is in 1995, and I'm going to show you here. Those are clinical trials for patients with the standard form of leukemia, which is the most common form of leukemia in children, the B-cell leukemia. Represent 85% of all leukemias. In the 1966, this is the first trial that was done. It enrolled 90 patients, and the survival rate was barely 10%. Today, this is where we're at, 96% survival rate. So there is a tremendous advance that has been made in the field of pediatric leukemia, and this is because we know better this enemy. We know better the disease, and knowing it better, we could target it better. And this is actually the power of clinical trials, because we change one thing at a time, and we reflected on the results, and we progressed with that. This doesn't say that leukemia is a done deal, and that's it, we can cure. This is the most standard form. There are other forms of leukemia that we're barely touching, similarly to the adult side. So what about brain tumor? Everybody knows glioblastoma, and unfortunately a recent event that happened to Senator Kennedy. Senator Kennedy died of a glioblastoma, which is one of the most aggressive forms of brain tumors. This is the most common form of brain tumor in adults. It happens in the elderly population most of the time. If you could say that elderly, of about 50, I'm coming to that age, so I'm soon to be elderly. And unfortunately, he wasn't here to see the beautiful signature. I'm sure all of you are aware that there was this signature for the healthcare that he battled a lot for, and he died of a glioblastoma. So glioblastoma is a very aggressive form of brain disease. That's the #1 killer today in brain cancer. And still, with a different means, and I'll go over that, we now know how to extend a little bit the lifespan of those patients. But unfortunately, we can't yet cure them. We will have the tools, and hopefully very soon. So in children, this cancer happens. It's not very frequent, however, it happens. And this is a brain MRI. This is a beautiful imaging study, if I can say beautiful, for a brain tumor that's very big, that's in the cerebrum, and that's very vascular. Anything that's white shows this is vasculature coming in here. So this is a mouse brain, and this is also to show you how imaging technologies are evolving. Everything that's yellow and green in here represent blood vessels that you could visualize. This is a brain of a mice where we implanted a tumor that's a glioblastoma. This tells you two things. First of all, we could image beautifully things, and this is happening even in patients because it's important to visualize this vasculature and visualize the effect of the treatment we're giving on this vasculature. And the other thing it serves to show you is that this glioblastoma tumor is amazingly vascular. It's absolutely full of blood vessels. And that actually makes it very, very hard to treat and makes it very invasive. The cells don't need to look far. They have found the mechanism to attract blood vessels. If you understood what Dr. Rak was saying, they could attract them, they could turn them to their own benefit, and use them to disseminate within the brain. And when you die of a glioblastoma, this is what it looks like. This is very necrotic, meaning cells that have died in areas, but also cells that are, this is a lot of blood vessels in here, and it's spread to the other side. We know a lot about the signatures, the molecule, the gene that you've heard of in adult glioblastoma. We know how it can come. We know a lot of targets. And one message that I would like you to know, because I'm going to use it, there is one target, one gene that's called the epidermal growth factor receptor that's amplified, meaning there are a lot of copies in it. And this is something that the cell uses to survive. And this is one of the drivers, not the major driver, but one of the drivers for this tumor. And why is that important? Because in children, we don't know anything. We know that they have this tumor, that if you show it to someone who analyzes the tumor, the pathologist, this person cannot distinguish between children and adults. So a lot of the treatment that are used in adults are being used in children for lack of better treatment, and the results are as disastrous, no survival, or very little survival. So our group tackled this. We asked ourselves, are glioblastoma in adult and children the same? And if they're not the same, can we identify targets that we could selectively go after in children? So we had a set of samples. We did what Peter said, a laser capture microdissection. This is a mouse brain. This is just to show you how powerful this technique. You could actually take exactly what you want to take and analyze it. And we took tumor cells that we analyzed. We took them from children and from adults, and we did, this is a graph where every dot is a tumor. And everything that is similar is going to cluster together. Everything that's different is going to cluster away. And we see, first of all, everything that's in blue is adult. Everything that's in yellow and red is children. That those are different tumors. And within the children, you have potentially, at the very least, two subsets. So the genes that drive this tumor are different between the adult and the children. And we know now that EGFR is not amplified in children. The genes that are upregulated, meaning you have a lot of expression of this specific oncogene in children, are different than adults. This is children, this is adult. And the ones that are downregulated, meaning that their expression is lower than when you compare it to a control brain, are different in adult and children. So with this information, we're able actually to identify several targets. One of them is called sortin Nextin 3. Why is this target interesting? Don't go, I'm not going to go into the physiology of this very complex stuff. What this gene actually does, what this protein, the product of this gene does, is each cell is made very wisely. We try and recycle everything. Like, when you recycle your paper, the cell recycles. It wants to keep as much energy for itself. It needs to renew. So when it gets a signal, the receptors that are at the surface over here, they go inside and they get degraded, and a little bit of that gets back up. So the signal comes, is for a very short time frame, and then it stops because it's degraded. So sorting Nexin 3, what it does is it tells, don't go to degradation, stay here, continue signaling, continue doing your beautiful work, we like you. So cancers like that, because instead of having a 1 second signal to grow, it will have 10, 15, 20, 30 seconds more, because the degradation that's natural after you get your signal is delayed. And this is what SNCS3 does. So if this is true, if you overexpress this gene, this sorting Nexin 3 in cell lines, you're going to have a growth advantage. This is what we call in our jargon soft agar, where you plot tumors that are transfected with nothing, this is the empty vector, or with your gene of interest, and in this case it's SNCS3. As you can see here, the one that had nothing don't grow well, here it is, but the one that have a little bit more of SNCS3, they love it, they grow. And when you do it in mice, the one that has nothing don't form any tumor in mice, but the one that have more sorting Nexin 3, SNCS3, form very vascular angry tumor in the mice. So this tells me that this gene, the fact that its level are increased, might potentially induce cancer formation and blood vessel arriving to a tumor, and might be of interest to pursue, and this is what we're doing. So what we think is happening is the major oncogene that have been described for adult glioblastoma, including this EGFR, this receptor that I told you about, are there, but they're not. Like in adults, what happens is your copy number, the number of copies of DNAs is increased. In children, you don't have that. But the cell found other mechanisms for this gene to do its function on a more prolonged basis. In adults, you have a lot of EGFR at the cell surface, and they signal because you have a lot of copies. In children, you have one copy, but this copy can work for longer time and do the same effect. So cancer across the lifespan. I hope I convinced you that cancers in children are different between adults, adolescent, and young children. You need to know one child in 450 children will have a cancer before the age of 15, and it's going to be mostly between the age of 1 to 5. There will be, as I told you, leukemias in 1/3 of the cases. Brain tumor is next, and other type of cancer are after. And what we know, based on what we see, is the cancer seems to be more aggressive. If you have the same cancer, it is going to be more aggressive in children. Maybe one way is because the vessels or the possibility to heal, to regenerate in young children is much better than in adults. This is a presumption. I don't know. It's an assumption. But this is one of the things that we need to consider. Just to give you a cliché, an example. If you have a fetus in the womb and you perform a surgery on the fetus will heal and you won't even know that this surgery was performed. The skin will have no scar. This tells you how strong our healing processes, how strong our regeneration potential are when we are young. We lose that. We keep some, but we lose it with time. We heal less and less. We have more problems. Our vessel age. So potentially this explains why, thankfully, in elderly patients, the cancer is probably not going to be very aggressive. We'll have a hard time spreading. However, in younger children, not the same hurdles. So spread is potentially more the rule than it is the exception. And the cancer stem cell, a lot of things are being done that I won't delve on it. What is a cancer stem cell? People believe that your cancer comes back because there is something that actually is at the origin of everything that you are not touching with your treatment. This hypothesis is the object of a lot of debate, and I'll be happy to take questions after that. So there are commonalities and a common core that could be exploited, but there are differences. You cannot simply do a cut and paste. If you have a target that works in adult cancer, before you apply it to pediatric, you need to prove that it goes. At the same time, for young adults and adolescents, maybe we know that they benefit from treatment that are tailored for children. So they should be treated potentially more like children than adults. So what about, I promised Michelle that I would touch base with you about targeted therapies. What are targeted therapies? Okay, we've spoken about all of those genes, all of those processes, all of the things. So we spoke about Avastin, the anti-VEGF. What is it doing out there? When you have a brain tumor, a glioblastoma, the first step that you do is see a good neurosurgeon, because removal of the tumor is key in your treatment. And this is what's done. In this case, this is the tumor. The neurosurgeon is equipped, and neurosurgery actually made all of surgeries with navigation, neuronavigation, intraoperative MRIs. You know exactly where you are. You know exactly where you could go. You could track, actually, some neurosurgeries are made with the patient wide awake. This way we know we're not touching the language center, we're not touching the motor function, and we can remove as much of the tumor without creating more harm to those patients. And this is what's done. Once you do that, the next step is radiation therapy. If you don't do it, it's going to come back. I hope I convinced you that based on the fact that they are so prone to blood vessels, this cancer, even if you remove it, has already disseminated in the adjacent normal brain, and you need to treat it aggressively. Radiation therapy is first, and then chemotherapy. Unfortunately for glioblastoma, after 4, 6, 1 year if you're lucky, it comes back. So what next? This is what's next. Bevacizumab is Avastin. There are a lot of trials, not only with Avastin, but also Avastin with other things, and I'll go back on that. This is a patient that had a relapse of his glioblastoma. As you see, this is a tumor in here. This is 2 different sequences of the same. Before treatment and after bevacizumab, and associated with a chemo that's called irinotecan. Beautiful response. This response, unfortunately, doesn't last long. However, it gives us more time. And when the tumor comes back, based on the little side effects that are induced by those drugs, this patient is still eligible for other trials. So we're buying time in this tumor. In other cancer, we're treating it. For breast cancer, Avastin is upfront for those that have spread to the lymph node, and the results are fascinating. The same for colorectal cancer. So this is a reality. It's happening. We're using them. We're integrating them into our treatment planification, and it's giving effect. Herceptin, you heard about it. What does it do? If you give it to a mice, what does it do? This is a normal vessel. When you have a tumor, your vasculature, as Janusz was saying, becomes very abnormal, very angry, it's there, and it's feeding this tumor that's here. This is a mouse brain. When you treat patient with a Herceptin, you normalize it. You normalize the vasculature, and if you go a little bit further, you make it inadequate, and the tumor shrinks. So this is what's happening. We spoke about Judah Folkman, so now, since the 1998, as Janusz was saying, for Herceptin, there were about 100 trials with anti-angiogenic drugs. We are now over more than 2,000 trials with anti-angiogenic, anti-proliferative, anti-adhesion molecules, and those are providing results. This is a slide. Knowing the enemy is, if you know your enemy, you could treat it better. I'm not going to ask you about that. This is how complex it could be. And it's even more complex than this. I wish it was as complex. Learning about it would be fine. But it's even more complex than that. But we are doing it. We are understanding more and more how cancer happens, how we can prevent it. And this is just an example of the phase 2 and phase 1 trials that are here with bevacizumab, with other anti-integrin, anti-angiogenic factors. So it is a reality today. Those drugs that were designed by people like Janusz, like Peter, like others, are now of benefit to patients. And this benefit is only going to increase. What we need now to understand is which population of patients can benefit from which combination of drug. Tailor. And I wanted to finish with a message of hope, because hope there is and hope is true. There are more and more cancers that we can prevent. We can prevent cervix cancer. How can we prevent it? This vaccine. We understand now that the HPV virus is actually the one that induces it, and we can prevent it by vaccinating those young women before they develop cervic cancer. We can prevent colorectal cancer by improving our diet, decreasing our weight, by doing those colonoscopies that everybody hates but detects those tumors. And this is true. There are more and more cancer we can treat. We're treating better and better colon cancer, breast cancer. And there are cancer where we are getting a little bit better at treating. Glioblastoma, potentially pancreatic cancer, most certainly kidney cancer. That was a death sentence a few years ago. And I'll end up by saying, I paraphrase, I like this. This is not the beginning. This is not the end. But for me, it is the beginning of the end. Thank you. So I'll take now my cat hat as a moderator. Peter and Janusz, do you mind coming here? And please, any questions, we're happy to take.
I'm not familiar with that trial. Do you have any additional information? Okay. Okay.
I could potentially. So, if you don't mind repeating the question, and if Dr. Siegel can't answer, I might have the beginning of.
You heard me.
Right.
For the audience, more than anything, it's fine.
I'd just like to comment on a drug study that they're doing now called the Braxen for a person who had breast cancer and sometime later had cancer that metastasized into the spinal and lung area.
Yeah.
Okay, I'll answer. So sorry. I hope I didn't hurt you. It wasn't actually part of my profile to go with that, so you'll need to bear up with me. So this is, a lot of trials are made with vaccines. So what they do is they take part of the tumor and they they take a little bit of blood from the individual and they try and create and generate T cells, immune cells that can recognize the tumor and go and kill it. So in Germany, for example, there are a lot of trials for glioblastoma, for breast cancer with that. When you look at the result that they are showing in some meeting, it looks astounding. It's beautiful. This is a cure. In reality, no. It works potentially for a small subset of patients, but it doesn't work. And why is that? We don't know. What I can tell you, I could give you an example for melanoma, which is another cancer where this type of immune therapy was very much done, where they took cells from the tumor, generated those immune systems, the vaccine stuff, and injected them. They worked up to a point, and they worked only when you gave full body radiation to the individual to kill his immune system. Why is that? Is that one of the problems in cancer is our own immune system creates kind of it actually tolerates the tumor. And whenever you give other cells that are designed to kill it, it just tells them to stop. And those, what we call the T regulator cells. And we need to stop them in order for the other cells to come. But that only works for a small time frame and doesn't work for all cancer. So I gave you a long answer for your question. Those trials exist. They're being developed even more for glioblastoma, adult and pediatric. The promise of working, I'm not sure. I think if you're very desperate, if you're out of option, you might consider, because this is what I have a family, one of my family who went there and didn't help, but this is not the reason why I'm saying that. The results are here. They help in the very minority of patients, and we don't know what helps.
Okay. I was told by a physiotherapist that a machine that they use called TENS for pain, it activates the metastasis. Is that true? And would you know of any other machines or techniques or food or whatever stuff that activate metastasis? Wow.
Certainly, I guess any sort of activity that has an effect on sort of bone quality and that relies on the function of these osteoblasts and osteoclasts that I was talking about in terms of the bone metastases, whether it stimulates their differentiation or their function, can have an impact on what tumor cells will do if they find themselves in that microenvironment. But I'm not aware of any studies that would link sort of the use of any of this type of equipment to a higher incidence of developing bone metastases. And certainly being able to exercise and keep fit and this sort of thing is going to be a positive thing rather than some of these anecdotal situations where it may have been linked to increased metastasis. But I'm not aware of any studies that would concretely link one to the other. Yeah, so I wouldn't be afraid of you know, physical exercise and using these types of equipment for fear that it might exacerbate the spread of breast cancer, for sure.
Thank you. There is a company in the Montreal region that sells a product called Isocal or Isocal that is supposed to modulate the immune system. And I've been approached to use it as a way of dealing with my cancer. And in addition to that, because I also have myasthenia gravis, where I try to reduce the immune system, the immune response, they claim that this will not reduce it, but it will modulate it. Have you heard of anything like that?
Do you want me to answer?
You want to try that?
No.
Okay. See, you could modulate the immune system. This is a guarantee, meaning you could suppress specifically some, optimize other. That Isocal is able to do that, I don't know Isocal. I know that not, up to now you could do it in the mice, you could do it in some specific situations, and those are drugs that are not without side effects. So I don't know what Isocal is by itself, but the modulation of the immune system is a possibility.
It's a whey extract. It's a protein.
Yeah, it's whey.
Immunoglobulin
okay.
from whey extract.
Okay, whey is something that has antioxidative potential and is used, for example, in prostate cancer, where oxidation is something that's very important. By itself, if it's not dangerous, you should take it. It's not gonna, if you want to try it, I don't see a downside to there is no proof out there. There is no scientific data to support anything. So this is where it's in a way your choice. That's, if it is an anti-whey, a lot of new diets and everything are based on this actually, and it might potentially be beneficial, but I wouldn't advocate taking something that's not proven.
So I think the involvement of the immune system, as Nada was saying, can go two ways, right? So you can have an anti-tumor response where you have mounting an immune response that will actually try to clear the tumor, but you can also have a modulation of that response where you dampen or create immune suppressive environment, and then the tumor actually will benefit from that transition sort of from an anti-tumor immune response to a pro-tumor immune response. So it can be quite complicated in terms of immunity and cancer and how that you can have that shift from what you initially think as an anti-tumor response, then that what your immune system would mount, versus in a number of situations, if an immunosuppressive environment is created, that can actually enhance tumor growth and tumor spread. So
I think as a general principle, it's probably wise to really consult these specific questions with your attending physician, because, I mean, there are certain things that are generalizable. So there's some data coming from clinical trials or otherwise, that could be sort of applied in more general terms. But in specific cases, this is nearly impossible. So one has to know exactly what the specific situation. And so, for example, I know some sad examples of people who had taken homeopathic drugs, or they've taken the route of so-called natural therapy to the exclusion of the. So I know somebody personally who had a relatively curable lymphoma. It was during the time I was in Toronto, and that person was advised not to take the prescribed chemotherapy protocol, and he actually followed that advice, and shortly thereafter, literally months afterwards, he essentially succumbed to his disease. And I'm frequently wondering, where would we be, if he actually did not take that route and actually stayed with his therapy that was prescribed, albeit unpleasant. But so I think in those kinds of situations, I think it's, we could probably comment on some of the general issues, but in terms of specific issues, I think it'd be very difficult, because this is only something that you and your physician may know. And if you need a second opinion, you probably should seek the second opinion. But I think in this specific context, it's very, very important.
Okay. If a patient survives one cancer and within 4 or 5 years develops a new cancer in another location without touching the first organ, do we assume automatically that we're talking about a metastasis situation, or could this be another primary cancer? And if that, either way, what are the differences in terms of therapy?
I think certainly that you can have the possibility of it being linked to the first primary, right? So this notion of the ability of these tumor cells to spread very early during the growth of the primary, and that you can have tumor cells disseminated to a number of different locations. Even after successful treatment of the primary, you could get relapses to different locations, and it could be as a result of the original primary. So that's certainly a possibility. And this sort of links into the idea of cancer dormancy. And in the case of breast cancer, you can have this situation where you've had a primary tumor, it's been successfully treated, and then you can have periods of 20 to 30 years where you actually have relapse in the absence of a second primary. And the idea will be, was that an early escaped tumor cell that was disseminated at the time that the primary was treated with, and now for whatever reason, whether there was an insult in that particular location, whether there was an inflammatory event or an injury or something that caused a reawakening of those tumor cells that were dormant there.
So I can conclude that we're always talking about a metastasis in that scenario.
Not necessarily.
Not necessarily.
You're right. Yeah.
Actually, as Dr. Siegel was saying, it might be the first cancer that had spread and wasn't detected, because there is no way we could detect it at that time. And when, even if you have, for example, for breast, no sign that in the breast anything came back, this other location where this cell went and woke one day for whatever reason, but it could be another type of cancer, and the only way potentially is either you had a blood marker, or more is actually to get a sample of it and to analyze it. Right. It is still treatable.
Right. So when they do the pathology, they can tell the difference? Usually they can. Yes. And so the treatments are different also?
Right.
Okay. All right. Thank you.
There was actually somebody before, and then it's you.
Just to follow up on the last question, then it really means that the child who has cancer, let's say leukemia, is really marked for life with the possibility of secondary cancers developing. There have been a number of articles in the popular press about this in the last 2 or 3 years, of heart issues, bone cancers, a variety of cancers developing on pediatric leukemia patients, which really means, in a sense, they're not cured with the 95% graph that you showed earlier in the evening.
There are two things. First of all, the 95% is for the most common form of leukemia, which is a B-cell leukemia. And even 95 for me is not 100, and there's still more that we could do. And the second thing, there are a lot of papers now, because now in a lot of cancers, we have turned children into survivors, and we have follow-up data. So out of those follow-up, and there's a flurry of recent articles, for leukemia there doesn't seem to be, for those that didn't get radiation therapy, and radiation therapy has now been completely removed from the schema of event since 1985, I would say. There is not an increased risk of secondary cancer. But for other type of cancer, like sarcomas or the brain tumors, there is an increased risk even of relapse at 9 and 11 years after. It's very sad that we cured or seemingly cured an osteosarcoma, for example, or an Ewing sarcoma, and they come back 9 to 11 years after. Whereas the premise initially was if they didn't come back in the first 3 to 4 years, they would never come back. So the data for leukemia is more encouraging for the lymphoblastic leukemia. Based on the current treatment modalities, we are not giving them other cancer. What we know now is that there are family disorders. For example, there are children with leukemia where you will find that the mother has breast cancer. And we know that there are genes within the family that are actually predisposing both cancers in happening. This is what we're learning more. So I don't know if I've answered your question. Leukemia is not a done deal. We can do more, and we have to do more. We need also when you are given the diagnosis of cancer for a family, for an individual, it's devastating. There are a lot of things, and this is something that you're going to be listening to in the next session, is how to deal with your life when you survive cancer. Those are major issues. We are, for children, for some of the cancer in children, we've turned them into survivors. Now we need to deal with all of the psychological effects, all of the side effects of this has brought to the patient and to his family. The lady over here, sorry.
A quick question about melanoma. It appears that it's a much more difficult cancer to treat, or it's more resistant to the drug?
It is very resistant to drugs, but there's some really encouraging new trends in melanoma field. So, for example, there's a new class of drugs that are called PARP inhibitors, whatever that means. It's not important, but which recently, within the last year or year and a half, emerged as very, relatively effective treatments for a specific subset of patients with melanoma. So really what's happening now is that these diseases that were defined rather broadly, traditionally based on histology, now I was saying about brain tumors, when you look at the tumor section under the microscope, they all seem the same. But within these different categories that have been defined, there are subcategories which are driven by distinct molecular machines. And in melanoma, it just so happens that subsets of patients are particularly susceptible to drug that targets one of these molecular machines. So yes, there is some progress, albeit very slow. And I think in melanoma, this is a very frustrating disease because 2% of all cases of malignant melanoma actually regress spontaneously without treatment. The remaining, there is a fraction of malignant melanomas which manifest themselves already as a metastatic disease. It's a so-called unknown primary type of disease. And thirdly, there are melanomas which metastasize subsequently and are very difficult to treat. And there's really no standard of care for melanoma as it stands now. The only standard of care that's known. So melanomas are treatable by surgical excision in the very early stage. But for metastatic melanoma, there's really no effective standard of care. There's one drug that is being used, DTIC, dacarbazine, and that's pretty much all to it. But there are new trends, new very promising trends just recently emerging.
Yes. Just also to elaborate on this answer, for example, in breast cancer, in all of the other epithelial cancer, you remove and you give adjuvant treatment, and you know that this is efficient. In melanoma, you remove And whatever adjuvant treatment you could give, even if it's very high, it won't affect. If the tumor has spread and if it has acquired the capacity to live elsewhere, that's it. You can't prevent it. You can't stop the one that went outside from awakening at any time.
Yes, I would like to know, is there any evidence that the zelandronic acid can prevent breast cancer from metastasizing to the bone?
So these are a class of drugs called bisphosphonates, and the way that they work is that they actually accumulate on the bone surface. And the cell type in one of my slides, which is an osteoclast, which is a cell type that resorbs bone, it internalizes the bisphosphonate that gets accumulated on the bone, and it actually kills the osteoclast. And so what you're doing is, again, you're sort of indirectly targeting the metastases by removing a cell type that's critical for those tumor cells to grow. So it's very indirect. And this really is the sort of the first line treatment right now for patients with osteolytic bone metastases. And that antibody that I was talking to you about, denosumab, is actually sort of following along that idea of trying to target the osteoclast. So one problem with bisphosphonates is that when you treat patients with bisphosphonates, that bisphosphonate will get layered down on all bones that are being remodeled. So you're getting an accumulation of bisphosphonates in the body over time. And you normally have normal skeletal remodeling throughout your life just to repair small defects in your skeleton. And if you're getting an accumulation of bisphosphonates, that can actually impair normal bone turnover that's a part of just normal renewal of the bone tissue. So what's interesting about this antibody is that it's targeting the same osteoclast by using an antibody that you can administer and that will persist in the body for a defined period of time. To actually try to accomplish the same thing. And these phase 1 and 2 clinical trials are indicating that this antibody against rank ligand is as effective as the current bisphosphonate treatments that are being used to manage bone metastases. So that's promising in that you won't sort of get these long-lasting, more chronic effects of that bisphosphonate on normal osteoclast behavior when it's just trying to remodel bone as a normal part of the physiology. Right.
As well,
mm-hmm.
Can be effective for both.
Right. So it's the same principle, right? So in osteoporosis, it's a process where you've tipped the balance to more osteoclast activity. Therefore, you're having more bone resorption than you have bone formation, and then you have the thinning of the bones and the fractures that are associated with that. And the antibody was actually first treated. This denosumab was first treated for osteoporosis. And then it was applied to sort of the cancer setting with those cancers that are inducing a sort of an osteolytic type of metastases or a bone resorbing type of metastases. Yeah. So right now, like the bisphosphonates are the first line treatment for patients with bone metastases. But now there's sort of these new drugs that are coming online now, which are sort of trying to accomplish the same thing, but trying to minimize sort of systemic effects that those bisphosphonates might be having.
There's
what's the name of the antibody?
Denosumab? Denosumab is it's Amgen that's taking that forward.
Okay. Go ahead.
I just wanted to say thank you to all three of you. Nice work, and I know you're tired. It's the end of the night, so thank you very much to everybody.
Are there other questions? There's one question over here.
This might be kind of a big question, and it's really me being overly curious perhaps because it's a little not completely directly what you're talking about, but in that the vein systems are so important, are there studies or do you know of studies where they've seen patients with either clotting factor issues or platelet issues or even allergies that seem to affect the way that metastases distribute or will or won't form?
Certainly. I think this is one of the things that really is coming to the surface in a rather major way, that clotting problems actually correlate relatively well with metastasis, to the point where anti-clotting agents are being introduced gingerly, I might say, but with certain determination into protocols of treatment in metastatic disease. And there are some really interesting data from large clinical trials with where anti-clotting agents are normally used for patient management, because cancers are usually associated with very high, 7 to 7 times higher risk of clotting than in the normal non-cancer population, even population which is at risk anyway for clotting, but cancer even makes this risk even greater. So for that reason, some of the anti-clotting agents like low molecular heparin, for example, or warfarin, or agents of that kind, they have been introduced as a sort of supportive therapy. And it turns out that unexpectedly to many, these agents turn out to deliver survival benefit in some of these larger trials. So in experimental settings, there is a very solid evidence that if you block clotting, you can actually block or inhibit a number of processes that are associated with malignancy, including formation of blood vessels and metastasis and tumor growth and so on and so forth. And this is, there's a couple of explanations for this. One is that clotting per se perturbs everything, makes tumor environment hypoxic. In brain tumors, for example, this is an interesting story, because although brain tumors are highly vascular, they are dense, loaded with vessels, but most or many of these vessels are actually not patent or not perfused. They don't carry blood because they're plugged up with clots. And these clots cause death of tumor cells, and death of tumor cells causes more vessels to grow, and the vessels plug up again, and on and on and on and on. So in brain tumors, is a very significant component of it. And so the other point of this, which I think many don't appreciate, is the fact that clotting system is actually delivers certain information to the cells. This is not just a sort of a piping device which plugs the holes in the pipes, but it is a system which talks to tumor cells and talks to blood vessel cells in a very particular way. So yeah, I mean, there's a lot of mileage in that particular area. You're absolutely right. This is a big question, but it is a good one. Thank you.
We're going to take 2 more questions just for time issue, so please go ahead.
Thank you.
Actually 3, because you had your hand raised before. So this gentleman over here, the lady over here, and then the lady at the back. I'm sorry, I'll have you run.
Since some drugs work for some people and they don't for others, are there any treatment initiatives where a patient's cancer could be biopsied and tested in situ against a variety of compounds prior to treatment beginning to see the true efficacy of that compound, and obviously in a very short period of time screen a variety of compounds, at least maybe not as a first choice, but at least as a, I hate to say it, a plan B?
Right. So I think that this is an excellent question, because I think this is where you're going to see the treatment of cancer going in terms of breast cancer in particular, where Janusz mentioned that more and more we're able to break down certain types of cancers into a variety of subtypes within the same disease. And breast cancer is a good example where it's been long appreciated by pathologists that there's very different types of breast cancers, but now molecularly we can break down those into distinct groups which are associated with particular gene expression profiles. And you've heard this said over and over again tonight. And those profiles can be prognostic in terms of if you have a certain profile, you're likely to do better or worse than a tumor of this subgroup. But I think then the next step to that will be sort of predictive signatures, where you can come in, you can biopsy a tumor, you will be able to have these signatures then that can be created before and after a treatment has been applied, and then ask how those signatures change. And whether we now will be able to move towards trying to devise predictive signatures to say if an individual comes in, they have this sort of a profile, we know that there's an X percentage of chance that they'll respond to this type of a treatment. And so sort of these response to therapy type of trials and trying to get that information before and after, I think will be sort of the direction that we head in.
So this currently isn't being practiced then?
Well, for breast cancer, there are actually 2 signatures that are making their way into sort of clinical utility. and they're more prognostic signatures. So they're helping to try to direct treatment options for a particular subtype of breast cancer, which are sort of ER-positive breast cancers, but they're Oncotype DX and Mammaprint. So these are two gene expression signatures that have been created, which they can be applied to the tumor material that the clinicians are seeing to help them try to devise whether this particular subgroup may not be able to do well just with an anti-estrogen therapy, but this is a type of person that we should use that plus chemotherapy, right? So we know that if they have this particular signature, an anti-estrogen therapy will likely work and will likely be successful, but if they have this type of a signature, the idea will be they have to be treated with tamoxifen plus a chemotherapy, right? So this is already starting, and these are being implemented now. But I think really the next generation will be sort of being able to get more information in terms of response to a particular treatment, sort of before and after, and asking if we see this signature up front, we'll be fairly confident to apply this treatment with a good outcome.
Yeah, this is very. Your question was also if we take the tumor, we test it with drugs, why can't we do it and then we know if it will work? Well, I can tell you if you take a tumor and you do drugs on it, it's going to respond. Because the tumor behavior and the way you could give and deliver those drugs are so easy to do, and it's not the way you could probably predict a response. The way we're trying to overcome this is to see, okay, this one that has this signature responded to this type of treatment but not to this one. This is what Dr. Siegel was saying, is using actually this gene signature to predict which treatment, if more treatment is needed, which treatment would work and which won't. And it's coming, this is how we're trying to tailor the treatment. Because unfortunately, just throwing drugs on a tumor is very artifactual, and we know that we've been very much surprised after that in the clinical setting.
Yeah, I just have a two-part question. First of all, based on what you were saying about the detection, there's currently no imaging technologies to detect the metastasis early enough. So is that correct statement? There is currently no methods to detect the metastatic cancer early enough. And secondly, also understanding more now how the process, how it works, and the treatment technology doesn't even matter. Does it really help the survival if it's detected a little bit earlier, a little bit later, when the treatment starts?
What I meant is we have a lot of new imaging technologies that helped us actually. The PET-CT, if you do, for example, a bone scan where you look at the activities in bone and you do a PET-CT, potentially the bone scan is going to tell you there is nothing and the PET scan is going to tell you there is many more. So there are many high-resolution imaging technologies that are out there. However, for dormancy, the way Dr. Rak and Dr. Siegel were saying is when you have 1, 2, 3, 5 cells, there is no way today to detect them. And we know that for a fact. For example, in women, always we take women, I'm sorry about that, with breast cancer. If you do a bone marrow aspirate on a woman with breast cancer, you will find in a lot of series that there are tumor cells over there. And we know for a fact that a lot of those women that have those cells won't develop metastasis. So finding them, seeing them, is not helping us. We need to know which ones at one point woke up, switched the angiogenic switch, and are going to develop metastasis. And this is where our tools are failing. We are trying to overcome imaging by blood markers. There is a lot of onus now, and it's part of the staging process, where we are taking blood to detect what we call in our jargon circulating tumor cells. And we're trying to see if, first of all, this is to detect them, second, to study them, and third, to assess efficiency of treatment. Because when we treat, we see that they decrease drastically in the blood. And we could potentially, I'm saying potentially because we're studying that, this is the onus of a lot of trials that are ongoing, if they come back, is the patient escaping treatment or at a higher risk of relapse? So imaging is something, and we're getting even better at it. But there are additional means in the blood that we are trying to tease to see if we could predict metastasis. What you need also to listen, we've told you about metastasis. We told you metastasis is highly inefficient, thankfully to us. Out of the million cells that are going to spread, potentially one is going to be successful in establishing. But it's this one that we need to understand and to prevent. The last question up there, and unfortunately that's it after.
Hi. Obviously, not all growths can be excised or biopsied, but presumably should be followed for any change. Is there a growth in a specific area of the body that should be excised regardless of whether it was negative, benign, or non-cancerous?
I'm not sure. There are some benign tumors. I'll take the breast yet again for an example. We know that if we palpate something in the breast, we do mammograms, and in young women we do MRIs and ultrasound. And those help guide us in knowing if those are potentially benign adenomas, there are adenolipomas that are over there, or is it something that has a potential of being cancerous, and then a biopsy is guided. So it's a case by case, and this is where having a referral to a team that knows exactly what they're doing is important.
Okay, thank you.
And that's it. Thank you.